Good evening. My name is Rosa-María Lidón, and my co-chair is Alessandro Sionis. It is a big pleasure to introduce Dr. Bernard Gersh. He is the perfect person to give the Magda Heras lecture, for two reasons. First of all, Dr. Gersh is an eminent cardiologist, professor of medicine at Mayo Clinic. His interest field is very wide and includes different cardiologist issues. He has more than 1,000 publications. The second reason is so important, or perhaps more than the first one. Dr. Gersh was a great friend of Magda Heras, and for us, it is wonderful. Now we have the big pleasure to listen to him. Thank you very much, Bernard, to be here.
Thank you, Rosa-María, and it's indeed a great honor for me to give the Magda Heras lecture. Magda, there were many sides to Magda. She was a superb cardiologist, a mentor, fine, great editor of Revista, a devoted wife to Mark Elias and mother to Anna, and devoted to her family, a very large family. I can't remember how many sisters, but there were many of them the last time I saw them. And she was a woman who was very small in stature with a huge heart. And those of you who knew her during the last few years of her illness were amazed by the courage and the dignity that she maintained. And like many people in this room, she was a great friend to you, as she was to myself and my wife. She died almost 4 years ago, I think, and we still miss her. And again, I thank you for the honor of giving the Magda Heras lecture. So I'm going to talk about future and innovative approaches to the management of chronic heart failure, many questions and many directions and unanswered questions. If you look at the evolution of therapy for heart failure, shown on the left is the present, and on the right over here will be the future. The present is 1986 to 2014, and this was really the era of improving hemodynamics, reducing workload, and neurohormonal inhibition with ACE inhibitors, ARBs, beta blockers, the development of aldosterone antagonists, the ARNI drugs or neprilysin inhibitors, ivabradine. And I think what's really important from a clinical aspect is these need to be titrated to guideline-directed target doses. And one of the great failings, I think, in clinical cardiology is we use these drugs, but we don't look at the doses that they were used in the trials. And this is an area for quality improvement, disease management programs, and performance improvement systems. So as we try and develop new drugs, we need to remember that we have to use the old drugs in the doses that were used in the trials. Again, between 1986 and 2014, was the development of non-pharmacologic therapies. I put a question mark next to CPAP because although the evidence was quite strong that this might work, the trials have not shown it. In fact, they have, if anything, have been to the contrary, and I know a great deal of work in sleep apnea has gone on in Spain. The ICD, CRT, assist devices, and cardiac transplantation. So where are we going to go in the future? I don't know. But I'm going to take you through a number of different approaches. I think one of the most exciting, perhaps, and promising is untangling the molecular genetic web and the identification of new therapeutic targets, understanding mitochondrial dysfunction and sarcomere contraction and relaxation, signaling calcium transport and myocardial energetics. I'll say a few words about pharmacogenomic profiling, and then close with some comments about cell repair, stem cell therapy, gene therapy, the artificial heart, and biomarker and hemodynamic monitoring. So to begin with the first of 4 clear directions, new pharmacologic approaches, new drugs, pharmacogenomic profiling, and hemodynamic and non-invasive monitoring. So there's a lot of interest in the use of natriuretic peptides, and the neprilysin inhibitor being an example, but a good question to ask, why are we developing new natriuretic peptides when BNP levels are already elevated in heart failure? Why do we need to push them up further? Well, the answer is that what we measure is the secretion by cardiomyocytes of large but unprocessed amounts of the poorly active prohormones, pro-ANP and pro-BNP, or fragments of the biologically active BNP 1-32. So future approaches are really to augment current signaling of ANP and BNP, and one of them is neprilysin inhibition, which was strikingly successful in the Paradigm trial. At Mayo Clinic, my colleague John Burnett has worked on the application of synthetic modified natriuretic peptides, and other approaches include inhibition of those factors that degrade the natriuretic peptides and trying to stimulate them through other means. Interesting, this trial, the guided trial, raises some questions about not using NT-proBNP as a diagnostic tool. We know it's effective, but the effect of NT-proBNP guided treatment in patients with heart failure and reduced ejection fraction. And this recent trial of almost 1,000 patients targeted patients to an NT-proBNP of less than 1,000 pg per ml, and the results of usual care with biomarker-guided therapy were no different, absolutely no different. And really what this trial brings out, if you look at changes in NT-proBNP levels, they were the same. And what this trial really does, it emphasizes again the benefits of optimal guideline-directed medical therapy. And if we use these drugs in guideline-directed doses, the results are as good as if we use them guided by NT-proBNP. And this will be one of several trials, and we'll see if the others show the same. Now, this is the Paradigm heart failure trial published New England Journal, angiotensin neprilysin inhibition versus enalapril in heart failure. But around about the same time was a very interesting article by Dr. Gene Brownwald, The Path to an Angiotensin Receptor Antagonist neprilysin inhibitor in the treatment of heart failure. And what he pointed out, it was almost 50 years from the identification of the angiotensin converting enzyme to the development of this drug. And this makes 2 points. First of all, 50 years is too long. But secondly, it does take time. And I think that I have a great interest in stem cells, and I know people like Paco Vilas have been extremely active. Maybe what we've had to realize is that maybe we were overtly optimistic that we could bring this to the clinical arena in 10 years, when this relatively simple drug took almost 50 years from the beginning of the angiotensin converting enzyme. So we and others need to have patience. It takes time. Now, this is a promising new drug. It's a drug or a molecule with the name of omecamtiv mecarbil. Now, what is interesting about it is it increases the calcium sensitivity of the myofilaments, and it prolongs the duration of systole. It does not increase calcium into the cell, and it does not have the same properties that an inotrope has. It prolongs contraction. Small trial from Dr. Cleland, and there is now a phase 3 trial ongoing. The drug is in yellow, placebo in orange, and if you look at the effect on systolic ejection time, it's almost immediate, and then this is the administration of the drug, and then after a period of time, 96 hours, everything gets back to normal. Stroke volume also goes up. And I think what's very attractive about this drug, it has no effect on oxygen consumption, and in this study, there was no change in heart rate and no change in end systolic volume, and a reduction in end systolic and end diastolic volume. So this is one of the new drugs on the horizon It's very attractive from a theoretical standpoint, as it is not an inotrope. Bendavir is a cellular free oxygen radical oxygen scavenger, and it was thought to be very promising in ischemia reperfusion injury. And as you know, many drugs have been tried for reperfusion injury and have failed. And in this animal experiment by Cloner, what you see is a burst of reactive oxygen species followed by myocyte death, shown in the control patients or animals. And this is with ischemia reperfusion injury, hypoxia, and then reoxygenation. And this is with Bendavir. It is very, very effective. And then it was used in the Embrace trial to prevent reperfusion injury, and it completely failed. But one of these rather interesting things that happen in science, in a similar animal model, but one that included animals in failure, it was shown to be promising, and this is one of several studies of the same drug, Bendavir, but this is in experimental heart failure. This is the effect on ejection fraction. Control is in yellow, end systolic volume shown here, and end ATP synthesis shown here. So one of a number of interesting drugs that are being developed for heart failure. Now, this is a classic paper from 1982 in the New England Journal of Medicine, and you may say, why am I showing this? This deals with a particular phenotype of dibrizoquine and metoprolol metabolism. Now, dibrizoquine is a drug related to one of the antihypertensives, and it is used as a marker of a number of studies of metabolic hydroxylation. And in patients in this study who are poor hydroxylators of dibrizoquine, these are the metoprolol levels given the same dose as patients who are extensive hydroxylators of dibrizoquine. So you can see that you could, using the same dose of metoprolol, the plasma concentration could be as high as 600 ng per ml versus 200. And obviously this has huge implications for therapy. And this was really, in a way, the pioneer study that led to, or stimulated interest in pharmacogenomic profiling. And this paper was 1982, and I would say that over the last 30 years, this has been a nice concept that's always bandied around, but it's been somewhat of what I'd call a promissory note. Hasn't really fulfilled its potential. It sounds good, and it makes sense, but we haven't really seen the benefits. But I think it's evolving, and it's a dynamic and progressive field, and with the impact of genome-wide association scans and pharmacogenomics and heart failure therapies, we will hear a lot more about this, as we have more recently with clopidogrel and ongoing trials. I think what's going to be key to understanding this, and I'm going to come back to this theme later in this talk, is we have to really understand the phenotype that we're dealing with. And there are going to be many different approaches, clinical and demographic, perhaps using big data and artificial intelligence, and then all the omics, metabolomics, genomics, transcription, and proteomics. So this continues to be an evolving field. To go back to the biomarkers, some of the greatest interest, I think, will be in the detection of latent disease. Now, probably 20 years ago at the Mayo Clinic, we published a New England Journal paper on dilated cardiomyopathy, and we suggested that maybe 12% or so of patients had a family history. And more recent studies have shown that this is probably as high as 25 to 30%. So where there's a great deal of interest, both in hypertrophic and dilated cardiomyopathy, is whether we can detect the pre-dilated cardiomyopathy phenotype in mutation-positive carriers. And can we identify these people before they get the disease and modify the disease very early on? And so there are a whole host of biomarkers reflecting a number of pathophysiological entities that are under evaluation, as well as other approaches, global longitudinal strain, CMR. Some of you know I work in South Africa. I've just seen some really interesting data that in HIV survivors who HIV-infected patients who are completely asymptomatic and are evaluated before treatment without any overt cardiac disease. If you look at subtle CMR changes, they already have cardiac abnormalities. So the question is, can you modify them and identify them and modify them very early on? And can we characterize these pathways in more detail? Excuse me. So the other area that's evolving is that of wireless PA hemodynamic monitoring. And heart failure, this is just one trial by Dr. Abraham of the CardioMEMS system. And you can see that in terms of cumulated heart failure-related hospitalizations, this is the control group, and this is the group treated with the MEMS system. And it's just a matter of time as technology evolves that this will become a more sophisticated area of technology. It's interesting, again, and my own interest in Africa, as you know, I come from Zambia and South Africa initially. It's interesting that in Africa, where there's so few referral hospitals and such a widespread burden of disease, one of the real approaches to disease management is going to be improved use of digital technology. And I can see the situation where a device like this can be implanted at a central hospital. The patient goes back to an outlying rural area and is monitored by a nursing station in the referral hospital. And I think the application of these digital technologies is going to be very interesting and certainly very topical. Now to get to the more sort of complex area, and that is untangling the molecular web. Understanding the phenotypes of the molecular mechanisms of contractile function, calcium signaling, myocardial energetics, and the role of microRNAs, and what will this lead to new targets and drugs. Now I'll show you this diagram for only one reason. These are the cardiomyocyte signaling pathways involved in left ventricular hypertrophy and heart failure. And as you look at this, one has to be amazed by the complexity and the sophistication of these multiple signaling mechanisms. What you also realize is there are many, many pathways. So if you block one pathway, there are many other pathways. There are many redundancies built into the system, much like a modern airliner. It's not going to depend on one part. And so what one realizes at first is, as sophisticated as this is, it's going to be very difficult to modify these multiple mechanisms. And this is the exterior. These are the insults, catecholamines, inflammation, reactive oxygen species. And these are the pathways all the way down into the myocyte that reflect, ultimately result in changes such as left ventricular hypertrophy and remodeling. And so one realizes that studying these pathways and blocking them is going to have very little effect when you can go down these other pathways. But what the interest now is to focus on the distal parts of these pathways. These are the HDAC pathways here and the microRNAs, and I'm going to blow them up for you just to point out that the HDACs, there are already small molecules being tested to inhibit the HDACs. So if you can inhibit at this point in time or this point in time, one might have a major effect on LVH and heart failure. And so the HDACs have distal effects on cardiac remodeling. They're both protective and pathophysiological, and they occur way downstream at the cell, at the myocyte. And then the other area is that of the microRNAs, that are major regulators of gene transcription underlying hypertrophy, remodeling, arrhythmias. And each microRNA can repress 20 to 200 genes. So if you can identify those microRNAs that are beneficial versus those that are adverse, one might have a major downstream effect. As targets of therapy, as I said, each one can repress 20 to 200 genes. There are now the ability to silence microRNAs in vivo with substances or molecules called antagomeres. And this is one of several microRNAs that improves cardiac contractility in the failing heart. And there are a number of knockout models. There is tremendous interest in the microRNAs for obvious reasons. The key I guess, to contraction and relaxation is calcium cycling and the mitochondria. This is a diagram that illustrates impaired normal mitochondrial function up here with normal ATP production, contraction, and relaxation. Here is a heart failure model with myofibers and fibrosis, depressed ATP, reduced contraction, and reduced relaxation. And in the normal heart, calcium cycling is absolutely, and in the abnormal heart, calcium cycling is absolutely critical to contraction and relaxation. Shown here is the sarcoplasmic reticulum, the cytoplasm, and the release of calcium through the ryanodine receptors into the cytoplasm is what enables troponin C to stimulate actin and myosin and contraction. And the reverse is the reuptake of calcium through the calcium pump, uptake pump, which is modified by a number of factors. But the re-entry of calcium into the cell and the sarcoplasmic reticulum is critical to relaxation. And the key mechanism really takes place here. This is phospholamban, which when phosphorylated, and when not phosphorylated, inhibits this calcium uptake pump, and when phosphorylated, it stimulates it and releases calcium. And there are a number of therapeutic targets, phospholamban, which, as I say, inhibits, but then when phosphorylated, stimulates, and a number of other calmodulin kinase. And there are a number of these potential therapeutic targets that can affect the calcium uptake pump. Irrespective, this mechanism is essential to the normal function of contraction and relaxation. And in fact, one of the downsides of ischemia is an effective ischemia on this pump in an adverse model. So this led to the CUPID trials of gene therapy in heart failure. The hypothesis was the deficiency of sarcoplasmic endoplasmic reticulum calcium ATPase, or SERCA2A, is associated with heart failure progression. And these are the references here. The CUPID pilot study of 39 patients using recombinant gene delivery of an adenovirus showed no safety signals, and this then led to the CUPID-2 phase 2b trial of 250 patients for 67 centers, and it was neutral for all clinical endpoints, completely neutral. So what are the questions? Is it inadequate gene transfer? Is it neutralizing antibodies? Is it the wrong dose? Is it the wrong agent? Was it the wrong target? What about the detrimental impact of sodium overload and reactive oxygen species that is part and parcel of the transfer of the gene therapy? And is it just early days? There's a lot to learn. And what we've now realized, and again, we come back to improved phenotyping, this paper by Hasenfuss, actually, which is from 1998, basically said not all patients have depressed activity of SERCA2A. These are patients with heart failure and reduced ejection fraction. Some do, and some don't. Maybe if we can identify who does, they will benefit from this therapy. But again, it emphasizes the complexity and how far we have to go. And I come back to this throughout the talk. We've got to understand the phenotype better. And now with the omics and other tools, we will understand the phenotype better. And heart failure is not one disease. It can't be. It's probably the final common denominator of many different pathophysiological processes. And one key to the identification and application of new therapies will be meticulous and detailed characterization and understanding of the many, many different phenotypes. And this is what role will big data and artificial intelligence play? Who knows? Another approach is an interesting one, and that is there is evidence that cardiac inflammation and can lead to fibrosis and heart failure. And this animal model is a recent European Heart Journal publication. What is depicted on the left is acute stress with beta-adrenergic stimulation interacting with reactive oxygen species here, forming a complex called the inflammasome. IL-18 is released, and IL-18 in turn goes on to cause initiation, inflammation, propagation, and ultimately fibrosis and cardiac dysfunction. And by being able to cleave IL-18, as shown here, the process was completely modified, and instead of ending up with fibrosis and inflammation and cardiac dysfunction, the animals had improved cardiac function. So interesting work as in atherosclerosis on inflammation. And then this is an important concept. It's mainly theoretical, and it deals with the concept of autophagy. Autophagy is a biological process used by cells to degrade cytoplasmic contents and remove damaged structures. And in some ways, it's been described as a housekeeping function. It's a good thing to have. And it helps maintain cardiomyocyte function and ventricular mass. And therefore, if one could understand it better and we could stimulate autophagy, this may have a profound effect. And it does apparently play a critical role in cellular remodeling and impairment of functions by age and disease. And it modifies the cellular response to environmental stresses by degrading these cytoplasmic contents. And that's all very well, but again, it's not so simple. Mitochondrial DNA that escapes from autophagy causes inflammation and heart failure, and in fact, in this interesting editorial, is it friend or is it foe? An attractive concept, but maybe, again, not so simple. The genetics of cardiomyopathy, I think, took a step forward in this work from the Seidman Laboratory in Boston, pointing out that the major gene in patients with familial dilated cardiomyopathy is the largest gene in the body, titin, and the truncations of titin cause dilated cardiomyopathy. Similar work from the United Kingdom show the same thing. The truncations in titin, the titin gene is the most common genetic cause of dilated cardiomyopathy in ambulant patients in the community. And I've now seen data that in a subset of patients with peripartum cardiomyopathy, titin may be involved. Very exciting breakthrough. And this is one of the exciting areas that I think will be the source of ongoing research. This is the classic paper by Mark Pfeffer, the diagram of acute myocardial infarction with infarct expansion leading to remodeling, chronic LV dilatation, and chronic heart failure. And this diagram was first shown here at the ESC, I think in Barcelona in about 1996, or even earlier, maybe 1992. And the idea of captopril and ACE inhibitors was to interfere with this process and prevent remodeling and chronic heart failure. And we have very exciting data that now that, and data in press, that we really have remodeling as we knew it is probably markedly diminished in the modern era. But where there's some interesting work is, are the people that remodel, do they have this genetic titin abnormality? And it remains to be seen. So a very interesting breakthrough. What about non-pharmacologic and surgical and mechanical approaches? If you look at surgical therapies for heart failure, they're logical. The STITCH trial was modestly positive. Viability testing remains under evaluation. Revascularization and surgical ventricular reconstruction, the so-called DOOR procedure, no benefit in the STITCH trial, but the problem is we don't see LV aneurysms anymore. They've gone in the reperfusion era. But in other parts of the world, like Eastern Europe, they do see classic LV aneurysms with a big bulge. And so surgical ventricular reconstruction may still be helpful in selected patients. What about mitral valve repair with and without devices? And this is just one such device called the ACORN device, placed to prevent remodeling. At the moment, we've seen no mortality benefit at all from repairing the mitral valve in patients with heart failure. It does improve remodeling, but it may be that our repairs are still inadequate. And what I think will be interesting is to look at the results of the ongoing trials of ischemic mitral regurgitation, both surgical and also percutaneous, with the MitraClip and other devices. They may tell us more about which patients with functional mitral regurgitation and heart failure will benefit from an approach on the mitral valve. But so far, fixing the mitral valve with the odd individual exceptions has not been of any benefit. And my own feeling is that surgery is most effective when directed at the cause of disease and not the consequence. And remember, functional mitral regurgitation is a disease of the ventricle, not the valve, as opposed to degenerative mitral valve disease. So we'll see what these trials, particularly these trials of percutaneous approaches, are going to tell us about the natural history of repairing the mitral valve. But I still think surgery, the best results with surgery will come when it's directed at the cause. So a stenotic valve, fix it, and they'll do well. Destination and bridge device therapies clearly will be employed earlier in heart failure. There's ongoing miniaturization and evaluation. Cardiac transplantation, particularly in the United States, I think you have a better, more favorable situation in Spain. We lack donors. We will continue to lack donors unless we repeal the seatbelt law and ban snowmobiles. That was a great source of donors, but we have a huge waiting list. Xenotransplantation, animal investigation is ongoing. The immunologic challenge is formidable. And the late Norman Shumway, the transplant surgeon, said, xenotransplantation is the future and always will be, and I suspect he's right. Now, we know about LV assist devices as destination therapy and bridge to transplantation, but this is really interesting, that about 1 to 20% of people, particularly with non-ischemic cardiomyopathy, actually demonstrate a recovery, quote recovery. I'll come back to that. I think it's closer to 1 to 5%. Despite that, by and large, we've been very reluctant to explant the devices. And what is interesting is that when you look at the studies, there's a real discordance between the functional improvement. The ventricle gets smaller, the ejection fraction goes up, but if one looks at the cellular and the genetic substrate, it hasn't changed. But I think the mechanisms are potentially fascinating. Is it unloading alone? Is there really cellular and structural recovery as well as functional recovery? Can we predict it? Can we augment it? Do we understand the molecular mechanisms? And will this ultimately lead to the identification of new therapeutic targets? The term I would use at the moment is not recovery. Because we've not seen the improvement in the cellular and genetic substrate, I think what we're looking at is a remission and not a recovery. And in fact, if you follow these patients, many of them will relapse at a later date. But there's a story here. And as we understand it, maybe we will identify new therapeutic targets. A few closing comments about cell repair therapy. The targets are very clear. You've just seen this diagram. This period of time here, this process is characterized by reduced vascularization and increased apoptosis. And the theory of stem cells is simple and beautiful. Adding stem cells can either cause regeneration, or as we've now seen, maybe through paracrine factors, we can get a modification of apoptosis and vascularization. And that was the theory, and what are the lessons that we've learned from the last 10 years? It's safe and feasible. Most of the studies have been done in bone marrow stem cells, mononuclears and hematopoietic. Skeletal myoblasts cause ventricular arrhythmias, and I do not believe that a skeletal myoblast will ever become a myocyte, and there's very little work in that area ongoing anymore. The results, a modest improvement in LV function, about a 3 to 4% ejection fraction change. Improved perfusion, remodeling, and coronary flow reserve after MI have been shown in some studies. All the trials were underpowered to answer clinical endpoints, but they all point in the same direction, and it's the right direction. It's an encouraging direction, but they are underpowered. The downside, 1 week after implanting the cells, they've all gone. there's no evidence at all of myocyte regeneration using existing therapies. And which I'll show in the next slide, the current theory is that there is a paracrine hypothesis as an explanation for the beneficial effects, because we do see beneficial effects, but we don't see the cells remain. And then there's the possibility of stimulation of endogenous cardiac progenitors, which I'll show you. Recent trials are mixed results. There are new questions have been generated. We don't really understand completely the basic mechanisms of benefit, but I can tell you there's extensive research ongoing and both into new cells and innovative strategies to try and improve the environment, which is a hostile one, so that the cells survive for longer. And if the new cells and the new strategies in fact work and improve the survival of these cells, we'll have to go back and look at all the safety issues, re-look at them. Because remember, a stem cell is a primitive cell. It doesn't always end up as a myocyte. It can end up as a tumor. The theory 10 years ago was that bone marrow cells would directly differentiate into endothelial progenitors or endothelial stem cells that would create new blood vessels. The other hope was that they would differentiate into bone marrow stem cells that would then regenerate myocardium. One theory at the moment is that bone marrow stem cells perhaps provide a stimulus to activate resident cardiac stem cells, which will go on and produce healthy myocardium. The most recent trial suggests that this is not the case. The paracrine hypothesis, however, is that some substances, molecules that are generated by these cells in culture may be angiogenic, with the ultimate production of new blood vessels. They could be cardioprotective, homeostatic stimuli, which may be immunomodulatory, and this is the so-called paracrine hypothesis. Now, why is it so fascinating? Because it raises the prospect of cell repair therapy without ever putting in the cells. Because if the mechanisms of benefit are through the generation of proteins and other paracrine molecules, maybe the future is you can grow these cells in a farm, in a fridge, take them out and just give the cell-free infiltrate or the cell-free plasma, which will contain these paracrine factors, and perhaps the cell-free cell repair therapy will be the key or a mechanism in the future. I think this is at the crossroads, to quote Sir Winston Churchill, is at the end of the beginning and not the beginning of the end. I like to think it's the end of the beginning. 2 years ago, I spoke at Paco Viras' meeting in Madrid, and I got it the wrong way around, and I said it's the beginning of the end. And since there were 500 people in the room who earned their living with stem cells, they didn't enjoy the beginning of my talk at all. There are grounds for optimism, ongoing basic research on multiple fronts, countries. Somehow it's been lost in clinical translation. The clinical trial results show safety and modest benefits. The clinical events are in the right direction. But what we really need are unequivocally positive phase 2 trials that then justify phase 3 trials. All our trials are small. Even the largest trial, the BAMI trial in Europe, it's small. It started off at 3,000 patients, which I felt was small, and it's going to end up around 600-800, I think. And we need really unequivocally positive trials. The concerns are these. The benefits are only modest. Many trials are neutral. I think that expectations of physicians and the public and the media have been unrealistic. That's partly our fault. So when you have such high expectations and you get neutral trial results, there's an overreaction in the other direction. Many scientists outside this field, in the scientific community, feel that stem cell research has been overfunded. In the state of California, it's $3 billion have gone into it. $2 billion raised by the state, and I think $1 billion by industry. And so people in other fields turn around and say, this is the amount of money you've spent and you really haven't got far enough. And maybe we should divert those funds elsewhere. It's a justifiable concern, to be honest. I think I understand it. I still think that this is a promising field, but I understand it. And then, unfortunately, there have been a number of justified concerns regarding scientific credibility. There have been 4 or 5 instances of well-known individuals where there has been fraud, and this has really seriously hurt the field. I also put here unjustified because there's been an article in the British Medical Journal that hinted at a number of fraudulent aspects of stem cell work, and that article was completely and utterly unjustified, completely. So right now, I think the field is active, robust, it's international, encompasses both bench and bedside, short of funding. There are at least 39 trials ongoing with about 1,700 patients looking at different cell sources, cell types, and different methods of delivery. So the conclusions are, I think a pivotal trial of stem cells which demonstrated a change in ejection fraction of, I'd put here 5 to 10%, will change clinical practice. You may remember, I don't know if Dr. Maggioni is in the audience today, but right now, but the pivotal GC1 trial that established streptokinase and reperfusion therapy as the new modality, the benefit on ejection fraction was 3%. The ACE inhibitor trials, 5%. Resident cardiac stem cells and what we do at the Mayo Clinic called guided mesenchymal stem cells are promising. This is a technique developed by Dr. Andrei Terzik, where mesenchymal cells are grown and directed to go down a cardiac myocyte pathway. We need larger trials to assess the relative benefits compared to bone marrow stem cells and other progenitors. We think these new cells are better. They look better in the lab, but we need the trials to show they're better. Equally important are ways of modifying the environment so as to make it more receptive to stem cells and prolong their survival. I think allogeneic cells are likely to be the predominant cell type because autologous cells, when we take the cells from the patient's own bone marrow, remember we're taking cells from sick patients with hypertension, diabetes, ischemic heart disease. So we're taking sick cells and we're giving them back to sick patients. If you can take cells from a healthy donor and providing there are no immunological downsides to that, that should be the most prominent form of therapy or the most promising. And I think the data show that this can be done and that the immunological challenge, at least in the early studies, is very minor. Evidence for the paracrine hypothesis is strengthening. The prospect of cell-free cell repair therapy is realistic. For surrogate endpoints, as we talked earlier today, blinding is essential. So I'll close with prevention. This cardiovascular health study in the United States of 21 years of follow-up looked at the contribution of major lifestyle risk factors for incident heart failure in older adults. And these were the so-called favorable risk factors that predicted a reduction in incident heart failure. Physical activity, modest alcohol intake, BMI of less than 30, and non-smoking. Interestingly enough, in this study, dietary patterns did not come out statistically significant. That does not mean it applies to other countries. And if you had none or one of these, the hazard ratio is 1.0. If you had 2 of these favorable predictors, then the incidence of heart failure after 20 years was about, what was this here, 0.8. If you adopted 3 of these healthy lifestyle factors, it was about 0.7. And if you had 4 or 5, the likelihood of developing heart failure at 21 years was 40% less. So as we look at the future, we need to come back and look at the present again. We've got good drugs that work, and we need to give them to guideline-directed doses. But we can prevent heart failure. It is not inevitable. The statement was made that everybody over 65 has diastolic dysfunction. That's nonsense. We've looked at it in our echo lab, and we find many healthy adults with normal diastolic function in their 70s and 80s. So we have to come back to prevention. So this is actually from Mark Twain, but I like The Unknown Trucker better. If you don't know where you're going, it doesn't matter what road you take. Well, I think we know where we want to go, but we may have to take many different roads to get there. And Franklin Delano Roosevelt, one of our great presidents, it is common sense to make a method and try it. If it fails, admit it frankly and try another. But above all, try something. And it's going to be a long and bumpy road. There will be many methods tried and many will fail. But ultimately, we will find some that work. And this is a comedian, W.C. Fields. If at first you don't succeed, try again, then quit. There's no point in being a damn fool about it. So I close and I thank you again for the honor of giving this lecture in honor of our great friend and colleague, Dr. Magda Heras. Thank you.
Thank you, Werner, for this excellent talk. And we didn't plan to have any questions because I think it's different. I mean, it's kind of difficult to make questions about the topic you have chosen, but I would like to make a final closing remark. And at our coffee break this afternoon, one of my colleagues commented me that he told me that, is that Bernard Gersh? And I said, oh yes, it's him. Oh, he just looks the same, and I think I saw him like 15 years ago. And now I was listening to you, and I was thinking maybe he looks the same because he has found a way of staying young by constantly asking himself questions. And so we have many cardiologists in training here in the audience, and besides the content of your talk, that was great. I think there is an inspirational point here, is that if you really want to stay young, you need to constantly be asking yourself questions and read. So my comment is this one, just you've seen this. It's difficult to make predictions about the future, of course, but you should try. So thanks a lot for giving this talk, Bernard. It's been really an honor and a pleasure.
Thank you.